All right, welcome to day 10 of Unit 3. We're going to continue our discussion on enzymes today. We're going to take a little bit of notes, just kind of a refresher and some other things that we're going to learn about enzymes. And then towards the end of class, we're going to do an activity in the elephant toothpaste lab. It's a little bit of a messy lab, but I'll kind of talk about it more later in this video to kind of give you a warning of things to avoid and things to look out for. Just a couple of reminders before we jump into the notes. We know that the enzyme activity lab that we did last time, that is now due. It is officially due in Canvas, so if you haven't turned that in, make sure you do that as soon as you can. And then the lab that we're going to do today, the elephant toothpaste lab, that's going to be due at the beginning of next class period. All right, so we're going to go ahead and jump right into the notes. You can follow along with the notes that you can download from the agenda today. But we're going to start off with just a little bit of a refresher. First I want to explain what our objectives are. We want to be able to explain how our factors such as pH, temperature, and concentration can affect enzyme function. So those are the things that are going to be discussed today and how they relate to the function of enzymes. So without further ado, let's do a little bit of a review. What you can do is you can pause this video if you'd like to kind of just do this on your own. Spend about 5 minutes to do a little refresher on enzymes and see if you can come up with these answers yourself. Otherwise, I'm just going to jump right into the answers. The first one says enzymes are a type of, and the answer there is protein. So they're a type of protein. The second point, enzymes catalyze reactions, which means they speed them up by lowering the activation energy. Okay. The third point says the molecule that a particular enzyme acts on is called the substrate. The fourth point, substrates fit into the active site of particular enzymes like a lock and key. This is called the lock and key model. So those are the main points here. I'll repeat the answers. Enzymes are a type of protein. They lower the activation energy. The particular enzyme they act on is the substrate. Substrates fit in an active site, and that is called the lock and key model. So hopefully you're able to get a lot of those answers right, and we're going to keep going and talk about what are some things that can affect how enzymes function. The first one, of course, is basically the concentration of substrate. So basically, the more concentrated the substrate, the more likely it is that the enzymes will encounter them. So the way I like to kind of picture this is that you could have enzymes present in a cell, but if the enzymes don't have anything to work with, then they're not doing their job. So it's similar to like if anyone that works in a job that requires supplies, like if, for instance, you work at a restaurant and you work in the kitchen, if you don't have food to cook your food with, like to make meals with, then you can't do your job, right? So basically, you need to have a concentration of substrate. But there could also be an instance where increasing the amount of substrate does not mean that you're going to be able to do more work. Okay, so over here in this diagram on the right, we have low substrates, so these enzymes are ready to do their work, and basically anytime a substrate comes, an enzyme will pick it up. Over here, we're picking up the amount of substrate, so we have more substrates, and each enzyme's able to find a substrate to do its job. But then over here, by increasing the amount of work, there's going to be a point to at which how fast these enzymes can do their job. So this graph on the left kind of reflects that as well. So you can kind of think of the y-axis as the amount of work being done or the rate of reaction, and then the amount of x is like the amount of orders being put in. So again, suppose this is like a kitchen. If you only have like 8 workers, then you could only do 8 meals at a time, right? So that you're going to peak at a specific instance right here where that's the amount of food you can output. And by just increasing the amount of orders, that doesn't necessarily mean you're going to be able to increase the amount of work. The amount of enzymes determines how much work will be done. So that's our first point that can determine just how fast enzymes can work. It's the concentration of the substrates. The second thing that can determine how fast enzymes work or how effective they are is the temperature, the ambient temperature of the enzymes themselves. Specific enzymes have an ideal temperature range, and like all proteins, excessive heat can denature them, causing them to lose their shape. So every single enzyme has a shape, and if you heat that enzyme up too much, then it's going to denature. So for instance, we have an egg here. You can see that there's eggs cooking. They're kind of screaming in pain as they're cooking. And that's because the proteins that are found in eggs are very rich in proteins. They are denaturing because of the heat, right? So the enzymes are losing their shape, they're changing their shape, and that actually can be visualized. Like the whites of the egg actually turn white, and then the yolk will solidify as well. And then once you denature a protein, there's no coming back. Like you've basically ruined the protein, you've ruined the enzyme, and you can't come back from that. And so there's a specific temperature range that they work at. Likewise, if you took one of these eggs and instead of cooking it, you put it in the freezer, you do pretty much the same thing. You would denature the proteins, and then the enzymes wouldn't be able to work again. So there's like an optimal range. You can see here, like the rate of reaction is the best rate at which the enzyme will work, and there's a specific temperature for each enzyme that tells it this is the best temperature that the enzyme will work at. Now, not all enzymes will work at similar temperature range. There's enzymes that work in very cold temperatures. Like if you think of animals that live in cold climates, like polar bears or penguins, they probably have enzymes that work better in colder climates. There's also animals that live in hotter climates, so they have enzymes that could work in hotter climates as well. So temperature is very specific for the enzymes, and each enzyme has its own range that it works best at. pH is a similar thing, just like temperature, where each enzyme has a specific pH that it wants to work at. And this is kind of a gross image here. What's happening here is that someone's, I guess, curdling milk. So this started off as milk, and you can see the separation of the cream and the water from the milk. And the cream actually curdled because they added what looks to be lemon juice or some type of acid. So if we put lemon juice in inside of milk, then it will curdle, and that's because the proteins again are denaturing. So there is a specific pH range that each enzyme likes, and again, each enzyme is going to have a different pH that it likes. Not all enzymes like to be right in the middle. Not all enzymes would want to have a pH of 7. There's enzymes in your stomach that prefer to have a pH of 2 because that's what your pH of your stomach acid is. There's enzymes that would prefer to be in a pH of 10. So it kind of varies on what kind of enzyme you have. But either way, if an enzyme is exposed to either really low or really high pH values, that's far from what it's used to, then the proteins will denature and the enzyme will no longer be able to work. Another thing that can kind of slow down the enzyme function is something called inhibition. And inhibition is when another molecule stops the substrate from binding to the enzyme by blocking the active site or changing the enzyme's shape. So basically, let's say we have this enzyme, this purple enzyme over here. It wants to work on this substrate. So it wants to form that lock and key model, the substrate binding to that enzyme. But instead, this blue molecule comes in because it also fits in this active site. That blue molecule will come in and it will interfere with the active site of the enzyme. And so when it comes in there, the substrate can't bind and the substrate will just have to flow away. That blue molecule is the competitive inhibitor. It's inhibiting the substrate from going in. So this is called competitive inhibition. And it's competitive because they're both trying to go for the active site. Now there's another form of inhibition called non-competitive inhibition. And this is where the substrate can't physically go into the active site because the active site has changed. If you take a look at the example of this enzyme here versus this enzyme over here, the arms around the active site kind of got bent over here on the enzyme. So in this case, the enzyme is unable to accept the substrate because of the shape of the active site got changed. Now, what changed the shape of the enzyme? It was actually this guy down here. And this thing, when it bound to the enzyme, this is a secondary site called the allosteric site. And this site is basically where if something binds to it, it will change the enzyme's shape. A lot of times this occurs naturally in the body, just kind of as like a mechanism to control how fast the enzyme is working or how much it's working. But sometimes it could be like a medication that someone takes that can control the enzymes, or sometimes it's like a poison or a toxin that an animal or plant releases that can stop enzymes from functioning. And that's true also with competitive inhibition. There's different forms where we can compete for either the active site or the allosteric site. But either way, the end goal for inhibition is to stop the substrate from meeting with the enzyme. All right, so what we're going to do now is work on an elephant toothpaste lab. Today we're going to do this lab virtually instead of in the classroom, so it actually works out that we're doing it in an online learning day. What I'd like for you to do today is open up this PDF in Canvas, and every person is going to be opening up and working individually to answer all of the questions in Canvas. Just a quick note about this lab. It is kind of a messy lab, so I'm actually kind of happy that we'll do this lab virtually. Just because it might be a little challenging for a substitute to have to help with all the cleanup. So we'll watch a video of this reaction happening. If you want to see this reaction in real life, it's a really easy reaction to set up. Just make sure you have parental consent. You basically just need a container like a plastic bottle and 3 ingredients that are readily available at home or at a grocery store, and they're not terribly expensive. So if you want to do this reaction at home, you're more than welcome to. But I'm going to go ahead and just show you what this lab looks like. If you go to Canvas, it's the Bio Unit 3 Elephant Toothpaste Lab. This is a summative grade. So what does that mean? That means it's worth as much as a 10-point quiz. So it's really easy to get those 10 points, but it's also really easy to mess up and not get any points if you overlook some of these questions. So what I'd like for you to do individually is just to be able to read some of this background information. Normally these are the materials that we would use in class, and this is the instructions. So if you want to do something like this at home, you can. You don't need a flask. Again, you could use like a plastic water bottle if you'd like, but the major ingredients are hydrogen peroxide, yeast, and dish soap. You can use food coloring if you'd like, and I would encourage you to do this like in a tray, or better yet, your kitchen sink if you want to do this at home. But again, you don't have to. It's just if you want to see this in real life, you can. This is for summative grade again, so we're going to do our little lab report, and each question has the major point values and all the things that I want you to include in all those things. So I want you to write down over here like a model of the reaction taking place. Here are the things I want you to include for that, and then here are some questions I'd like for you to answer afterwards. So this is the agenda for today's online learning day. But without further ado, what we're going to do now is watch the video of the reaction, and then after that, we will go ahead and get sent off to do the lab report on our own. Alright, so here's the setup for the elephant toothpaste experiment. First and foremost, you want to have a clean working area, and safety first. You want to make sure you're wearing gloves and safety goggles if you have it. Make sure you have parental supervision, or at least parental consent that you're doing this experiment. And this is how you set up the whole experiment. You want to make sure you have an area that you're working in, like a pan or like a sink, just because, again, it's a messy experiment. A water bottle, a cup to stir like the yeast solution, and then like a stirring stick or a spoon. And then they call this solution here developer, but it's hydrogen peroxide. Then we have active yeast, warm water, and dish soap. And then an option is food coloring if you just want to make it colorful. And so here's the experiment. The experimenter in this video is going to put the main reaction area in the tub. That's going to be the water bottle, and pour in the hydrogen peroxide. So hydrogen peroxide, again, readily available at most grocery stores and pharmacies, but it can be an irritant, so you want to be careful with that. And this is where you would want to put in your food coloring. If you want to add food coloring, again, completely optional. It's not going to affect the experiment at all. Mix it around a bit to get the colors swirled around. And now you can add a little bit of dish soap to the whole solution. You can see here that the experimenter is not really measuring how much dish soap to put in. They're kind of spilling it everywhere. You want to kind of be accurate with how much dish soap you want to put in. So in the PDF, I say how much to add. And then you can actually make an observation to check the temperature. It should be around room temperature, so it's not going to be very warm. But then you have warm water in a cup, and then you put a packet of yeast to dissolve it into the warm water. And then you stir that around, kind of let the yeast just mix in with the warm water, kind of create this yeast solution. Then you're going to pour the yeast mixture into the bottle, and then you can observe the reaction afterwards. And so this is the whole elephant toothpaste experiment. So here the observer says that the temperature went up to 49 degrees Celsius, and that's pretty warm. That's like over 100 degrees Fahrenheit. So it's like maybe 110 degrees Fahrenheit or so. Um, and you can see it does make quite a bit of a mess. The food coloring didn't really do anything in this experiment. It's not really useful, but a couple of notes. Obviously, it made a big mess. The temperature went up, and the reaction happened very quickly. And then you can use this information to answer the questions in the lab report.